Good morning. This is the presentation about my internship in conservation biology that I completed at the Saving Turtles at Risk Today program in Orillia, Ontario. The Saving Turtles at Risk Today program, the START program, as I'll refer to it, first came to my attention when Scales Nature Park visited my herpetology course last fall. They came into our class, they brought all kinds of live reptiles, and they explained some of the fieldwork that they do through the START program. And it sounded amazing. They made it sound like you go out into wetlands, you're a total renegade, you get to be outside, and you are capturing wild animals for research purposes. So it sounded like something that was right up my alley. Now, Scales Nature Park, the people who came to my class, are tightly linked with the START project. The START project runs out of Scales, and also on the same premises, there's the Georgian Bay Turtle Hospital. So they're a real powerhouse in terms of turtle conservation, just at the southern part of the Muskokas. So they have a lot of connections, and there's a lot going on there turtle-wise. But the START project technically was who I was doing my internship with. So the research problem we were trying to address is a really fun, big one, which is that data sets and long-term studies are lacking in terms of turtle conservation in Ontario, and that range maps are often out of date. We'll find out why this is really important in a second. Since all of our turtles are on their way to becoming species at risk, they're a high conservation priority. This work is also made possible by private funding as well as public. Many grants have been given to start to do this work, and that might be due to the fact that turtles are such a charismatic and well-liked animal. Also, large swaths of the Muskokas remain unsurveyed, and development is accelerating. So the Muskokas are getting busier and busier all the time, and if there are habitats we don't know are being used, we can't protect them. In terms of turtle conservation, even just finding a turtle can be enough to protect habitat. So for example, if you were to catch a Blanding's turtle, like this happy guy, you get to draw a circle around that turtle of a 2-kilometer radius. All of those wetlands are now protected. That doesn't mean development can't happen there, but it does put a hurdle in the way of developers, and it allows us to make better choices about land use in that area. So every Blanding's turtle is like a treasure chest that you found. So it's like finding treasure, and we were constantly going out and trying to find Blanding's turtles in blank spots. So it was good to find the same ones again, but it was especially good to find Blanding's turtles in places they weren't known from previously. My goals going into the placement, I wanted to gain field skills in herpetology. That's what I'd like to specialize in, and there's a real catch-22 about it, where you are expected to have these skills before you're given an entry-level job. So volunteering is essentially mandatory, and so I wanted to get all the field skills that I could put on my resume. I also wanted to demonstrate ability to work outdoors, work in all weather. That's something that herpetology master's degrees and entry-level jobs are often looking for and looking for proof of. I also wanted to feel more comfortable in wetlands. They're a strange habitat for humans to navigate. Being in muck and cattails isn't a natural thing, so I wanted to learn my way around and to manage data and add that onto my resume as well. So here's the team. This is the Turtle Boot Camp crew. So Turtle Boot Camp was the first 2 weeks of my placement. They run it every year out of Scales for START, and you spend in an intensive 2 weeks learning and then getting out into the field. It's early in the season, which is really important for turtles, that we have such a large body of people out there, because early in the season is when you're going to find your spotted turtles, and a lot of Blandings are up basking as well. So it's a good time to get those really super key species. Spotted and Blandings were the main focus of our study in terms of fieldwork. So we spent one day in the classroom. That night we all had to have a wetsuit ready for the morning, and we went out into the field and we began to explore wetlands and to capture turtles. So it was very exciting and quick pace. So in essence, we were out in the field capturing turtles by hand. So that meant stalking turtles, not my specialty. Essentially, you're in the water up to here, and you have a buff over your face and you're hidden and you're sneaking up on turtles on logs. I did a lot more smash and grab, which was just to run up and run from pool to pool, maybe running across a bog mat and hoping that there's a turtle there who's surprised by you and you can grab them before they get away. It was a real thrill. So you can see in this picture, this was my best haul of turtles. This is 2 spotted turtles and a juvenile blandings. And I almost had a heart attack. It was so exciting. So it's really great and a lot of fun to do. But that's not where the work ends, actually. So once you have the turtle in hand, there's a lot that needs to be done to make that catch significant or to make it matter. So we collected data right out on the bog mat. So the START program has a setup where they have tablets and you're inputting data into their database right out in the field in the middle of nowhere. And so here's a juvenile snapping turtle, and for each species, it was a little bit different. So for blandings and spotted turtles, we would take a blood sample out of the tail, just a small blood sample. And for all the turtle species, we would measure their carapace, the top of their shell, their plastron, the bottom of the shell in various ways, the height of the turtle. Also on snapping turtles, we would measure pre-cloacal distance, so the distance from the plastron to the cloaca, which helps to determine their sex when you compare it to the last half of the plastron. So a further out cloaca means a male. So we did have to determine the sex of each turtle. If it was an adult turtle, we determined gravidity, so you would feel for eggs by placing your pinkies inside the shell. And we collected, various bits of data, including really accurate locations, ambient temperature, behavior when they were found, etc. So there was a form we were filling in for each turtle. We worked in really challenging conditions. The bog mat can be really tricky to get around on, and you're also having to set up a little data collection station on a sinking bog mat, which was interesting at times. There were also infinite black flies and mosquitoes, but it was worth it. It was really an adrenaline rush to be out there and to be doing something so cool and new and hands-on. So some of the most exciting things happening at START are that they found a new population of spotted turtles. I don't take credit for it. I was on the trip when the population was confirmed. We hiked for about 3 hours from the road, over bog mats and through rough reed marshes, but we did find a second spotted turtle in that location ever. So we know there is a population there, and work will continue to figure out exactly which habitats they're using. We also all signed confidentiality agreements at the beginning of Turtle Boot Camp because spotted turtles are such a species at risk. Their locations can't be disclosed, and they're not available to the public, and we're expected to also maintain that level of protection for them. Each turtle, also, you can see on this turtle, received a notch code. So you can see in the edges of the shell that we filed triangles into it. That's a unique code for each turtle, and that allows us to actually create really valuable mark recapture data. So each turtle has a code that includes their behavior, the last time they were caught, and the date. And each shell is also photographed like this, and also on the top. The turtle is still in there. So that we can eventually maybe do face recognition technology with the shells. That would speed up processes, and if we didn't have to notch turtles, that would be great. But for now, everyone is notched with a unique code so that we can catch them again and again and know who they are. And that data is especially valuable. So it allows us to have repeated captures with accurate coordinates, which allows us to infer their range, how far they moved, nesting migration, how far did they move to nest, what parts of the landscape are they using. It also allows us to measure things like growth rates, how their body condition changes, what parasites they have, and how that might be affecting them. If they're gravid, it allows us to learn about different gravidity times throughout the year, or how long they might be gravid. So repeatedly capturing these turtles is hugely valuable and increases the value of our data set a lot. So we visit the same sites over and over and over, even throughout the season, and also ideally throughout the years. We collected those blood samples for DNA analysis. We want to create family trees, especially the spotted and Blanding's turtles, and determine their breeding success and breeding ranges. So if turtle A is mating with turtle B and they're about 3 kilometers apart, we can know that the wetland habitats in between there might be a really especially important corridor for DNA exchange. So that would be something wonderful to be able to do with that data. The project also works with the Ontario Reptile and Amphibian Atlas, the best app in the world. I love that app. We record all of the species at risk and all of the herpetiles we encounter in general. So here's a smiling ribbon snake. All of our ribbon snakes were recorded, adding to the protection for the wetlands we saw. An eastern spotted newt and a water snake. These are not currently species at risk, but we record every herpetile because it's not known when exactly they will become a species at risk. But it's expected that they might at some point. So we're collecting a slew of data out there. When it gets hotter out, the turtles stop basking on the logs as much, and we started nesting surveys. We were driving around at night looking for nesting turtles. Here's a female snapping turtle thinking about laying a nest. We would wait. We would process the turtle as we did the others. We would also excavate the nest, and so that the eggs could be incubated. So that was another slew of training that we received to be able to do this process as well. We also documented roadkill of all herpetiles, including snakes and frogs, and on bicycle and in vehicles and on foot surveys, and we also documented predated nests. So if you ever see a turtle nest like this with the eggs above the ground, you can know for sure that they were eaten, because if the turtles were to hatch normally, they wouldn't bring their nests up. So all this work is made possible because Scales Nature Park does the outreach and education, START runs the fieldwork and research, and the Turtle Hospital, which is still up and coming, will allow rehabilitation. So this makes them a hub for turtle activity in the area and allows them to do outreach like this. So they've posted these signs. People in the Muskokas can report turtles. People in the Muskokas, if they're there for a cottage, generally they're pretty turtle friendly. They're happy to have turtles around. They want to do something, feel good while they're out there. So if they report a turtle, whether it's dead or alive or nesting, we can be involved in that and do the science required every time a turtle's spotted. So unexpectedly, I was able to do some great networking. I become a passionate advocate for turtles in my other jobs. Right now I'm a naturalist and I'm constantly harping on turtles to kids, but it's been a really great tool. I've been able to use the information I've learned. I'm comfortable in wetlands now, which is a really fun thing, one of my hobbies, going herping outside. And my career goals are much clearer. I know that herpetiles are something I want to get into. I think it's going to be snakes instead of turtles if I can manage it. But overall, it was an excellent experience. Thank you for listening.